This video explores sequence alignment in bioinformatics, covering concepts, algorithms, scoring, and applications. Sequence alignment is a method used to compare DNA, RNA, or protein sequences to understand biological relationships. Sequence alignment is a bioinformatics method to compare sequences, identifying similarities and differences, and helping us understand life. The two main approaches are reference alignment and de novo assembly. Sequence alignment compares biological sequences to detect similarities and differences. This method is fundamental for functional prediction, evolutionary study, and sequence analysis. The basic idea is to compare sequences to identify similarities and differences. We use pairwise alignment for 2 sequences and multiple alignment for more than 2. Sequence alignment helps us find similarities and differences. The two main approaches are reference alignment using a known sequence and de novo assembly without a reference. Sequence alignment compares biological sequences. Key methods include pairwise and multiple sequence alignment. ClustalW and ClustalOmega are popular tools for multiple sequence alignment. Global and local alignment are used to compare sequences, with global alignment focusing on the entire length and local alignment finding the best matching regions. Gaps and gap penalties are crucial for maximizing similarity. Sequence alignment is essential for identifying similarities and differences. Pairwise alignment compares 2 sequences, while multiple alignment compares more. The ClustalW algorithm uses scores and a guide tree for progressive alignment. Sequence alignment compares biological sequences and helps identify similarities. Key concepts include pairwise and multiple alignment, global and local alignment, and scoring matrices for statistical significance. Gap penalties are also important. Sequence alignment compares DNA, RNA, or protein sequences. Scoring matrices, identity, similarity, and homology help interpret the results. Tools like BLAST and LAALIGN facilitate efficient comparison and detection of biological relationships.